Thank you, Sarah, for that kind introduction. From Azim and Robin about Anne's contributions to science and to embryology, but as I think most of us will know, another of Anne's major contributions, and especially for those of us working in the area of bioethics and law, was, as Sarah Norcross has just pointed out in the chat, the role in establishing the 14-day rule in embryo research regulation. And so I'm extremely honoured to be invited to give this talk. And I think that at the point where we are now, in 2021, now is precisely the time in which we should be reflecting on not only Anne's scientific, but also her regulatory and ethical legacy and where that might go for tomorrow. So I've somewhat whimsically titled my talk Hopeful Monsters, and those of you who are evolutionary biologists will recognize this term, obviously, but the monsters that I'm thinking of, the hopeful monsters, are not mutations from evolution, but instead the ethical and the scientific imaginaries that we are increasingly in a position to create. So we know a few things are true, if you like, in the ethics and the regulation of human reproduction and embryo research. There are certain bright lines that from this point, from the early days up till this point, have held true. And these include, of course, the 14-day rule, that embryos should not be grown in culture beyond 14 days. And until very recently, the human germline itself was seen as a bright line, the idea that heritable modifications, and not only research modification of cells that would never be destined to become children, but heritable modification, the creation of genetically modified human beings, was the other bright line that we thought could perhaps never be crossed. But of course, current developments in basic science and in innovation are challenging these. So we have now new possibilities for what can be done with embryos. We have, of course, genome editing, and we have the possibilities of extended in vitro development that I'm looking forward to hear more about later. We also have developments in the nature of embryos. So we now have the possibility that we can create embryos from synthetic or in vitro gametes. And further, we have the possibility of creating synthetic, what has been called synthetic embryo-like entities. So agglomerations of pluripotent cells that begin to behave like embryos. So the question then is, as the nature of embryos and the possibilities for embryos are changing, how can and how should we revisit the way in which we regulate? Embryos and reproduction used to be a simple process. So we knew that sperm and eggs could combine to form an embryo, and then that embryo would either become a child, or perhaps if you are unlucky, it wouldn't. So embryos were very much about reproductive futures. And through the process of scientific development, we reached first a point where embryos not only gave rise to children, but gave rise to a set of choices about possible children through the application of pre-implantation genetic testing. In the era of human embryonic stem cell research, embryos also became a resource for research. With the development of not only stem cells, but induced pluripotent stem cells and what could be done with these, we've reached a point where, in fact, pluripotent stem cells can also become gametes, which can become new embryos. And as I said, with the generation of synthetic embryo-like entities, pluripotent stem cells might form embryos directly. Embryos themselves can recombine in different ways, for example, through the process of pronuclear transfer as used in mitochondrial replacement, meaning that we can intervene on the process of embryonic development in research. So embryos, from being a simple and well-characterized, have now become multiple, hybrid, and uncertain. So the possibilities of the embryo require us to re-examine the ethical arguments underpinning our regulation and how these may have shifted with the new technological developments. The early debates over the moral status of the embryo had the difficult, almost impossible, we might say, task of reconciling two opposing positions. At one end of the spectrum were those who thought that the embryo had what we might call full moral status, that human embryos deserved protection equal to that of born humans, and therefore their use, their destruction in research was impermissible. At the other end of the spectrum, those who thought that the embryo had no moral status, therefore needed no special protection, and held that destructive uses and research were permitted. So these two positions have often been characterized at opposite ends of the spectrum, with what's often called the gradualist view in the middle, which is the idea that embryos at the early stages may not have moral status, but that they acquire it at some point during development. So they gradually acquire moral status up to the point where obviously born humans have what we would call moral status. Now, it's often said that our regulation as a compromise between these two ends of the spectrum reflects this gradualist position. But in fact, that's not quite the case. The way in which we regulate the embryo, the way in which our regulation characterizes embryos, doesn't place them at some point along this spectrum of acquiring full moral status, but instead places them in a different category altogether. So embryos are seen not as having moral status partway between none and full human, but instead a different type of what's been called special status. Special status of the embryo requires respect, but it's not moral status of a sort that would not be compatible with destruction. So the special status of the embryo and the way in which we respect that special status is still compatible with destructive uses in research or with destruction after the use of other embryos in IVF, etc. And we can see from the introduction of the 14-day rule that in fact, the kind of respect that special status requires is that embryos be destroyed before what they might become. So it requires that embryos be destroyed before they might become either something with moral status or perhaps something the existence of which might threaten the present moral order. And this is why I call it the paradox of the status of the embryo, that in order to respect that status, we must stop it from becoming. Katie McMillan has written about the human embryo in regulation as a liminal entity, something that is in the process of becoming, and has characterized the ways in which our current regulation fixes the embryo and stops the processes of becoming which might be open to it. I think that the evolution of the regulation of reproduction in embryos gives us some clues to, if you like, this puzzle about the nature, or in fact the natures, the changing natures, not just of the embryo singular, but of embryos multiple, of the different things that embryos can represent, of the different entities that embryos might become. The 14-day rule, and in fact human embryos themselves, I would argue function as what sociologists have sometimes described as boundary objects. So both the 14-day rule and the human embryos are many things to many people. They enable discourse amongst those with different perspectives. And in this role, in the original discussions over the regulation of embryo research through which the 14-day rule was founded, they served this role in order to enable discussion to happen, led to the process of, through what Sarah Franklin has called biological translation, the implementation of the 14-day rule and the regulatory system that we have today founded upon it. So embryos in this multiple sense, embryos in this sense of being many things to many people, represent a multitude of different possibilities. They represent reproductive futures for would-be parents, as repositories of reproductive choice and responsibility. So if we look at the very much an argument about the reproductive choices that, along with that came arguments about the responsibilities of parents to make good or wise reproductive choices. Later in the stem cell era, embryos became characterized as resources both for research and for therapy. So along with arguments such as the nothing is lost principle, Harris has described it, the principle of waste avoidance. The fact that the IVF industry that had been enabled by the initial regulation that followed from the Warnock report had now resulted in the availability of a large number of so-called spare IVF embryos meant that an ethical argument could be made about how best to use those resources for research and for therapy. And it's important to note that in the stem cell era, as in the era of genome editing today, the therapeutic promise of the technologies has been entangled with the value of the research. So the idea both that stem cell research was necessary in order to realize the promise of regenerative therapies, the idea that genome editing research is necessary to realize the promise of therapies or of treatments, whether for infertility or for genetic disease. So the question, I think, is which sorts of discourses are being enabled by the different visions of embryos, both that have emerged over the past 40 plus years and that are now emerging with the new technologies of genome editing, synthetic gametes, and so forth that are available to us. The new visions of embryos that these technologies are opening up include new forms of reproductive future and new choices. So, for example, the technologies that have been used for so-called mitochondrial replacement therapy of maternal spindle transfer and pronuclear transfer have also been proposed as a means of enabling same-sex reproduction, so two partners of the same sex both to become genetic parents of a child. In vitro gametogenesis, the production of synthetic gametes, likewise has been suggested as a way of enabling new family forms with same-sex parenting. Embryos today also represent new routes to therapy. So we might look, for example, at the discourse over cytoplasmic hybrid embryos that took place around the 2008 amendments to the Human Fertilization and Embryology Act. And the idea that cytoplasmic hybrid embryos could represent a solution to the production of patient-specific stem cells via nuclear transfer that was soon overtaken, of course, by the generation of induced pluripotent stem cells. Human-animal chimeras and chimeric embryos, likewise, through the process of blastocyst complementation, are being suggested as providing yet another route to therapy by the production of, for example, human organs for transplantation. Reproductive genome editing occupies an odd position within this therapeutic space because, as the Nuffield Council on Bioethics argued in their 2018 report, we can't really conceive of heritable genome editing necessarily as a therapy for the child to be born. Instead, genome editing in a reproductive context is enabling, again, new reproductive choices for parents who might not otherwise be able to have children free from genetic disease. Now, we might conceptualize, and this is beyond the scope of what I'm going to talk about today, but I think increasingly, as we think about genetic diseases as shared amongst families and as affecting reproductive choice, it might be that we come to conceptualize, in fact, genetic disease as not only being about the symptomatic effects on bodies, but in fact about the disease of compromised reproductive choices. And in that sense, heritable genome editing might represent a therapy for the prospective parents. Beyond this, embryos also represent new imaginaries of future humans or future humanity. And so the rather futuristic discourse, for example, around moral obligations to genome edit in order to produce these new breeds of humans can be seen as a representation of these future imaginaries of the human. So again, the question I think that we need to confront is what sorts of futures, acceptable or unacceptable, these different embryonic natures and visions that are being enabled by technology, what futures do these entail? And I want to look now at the most recent iteration of regulation of embryos and research to examine this. So the 2008 Act that amended the 1990 Human Fertilization and Embryology Act introduced a new concept, where before we had had the embryo and the thinking around the pre-embryo that enabled the 14-day rule to be brought in. In the 2008 amendments, we introduced the concept of the permitted and the non-permitted embryos. And this, I suggest, is perhaps presenting us with a new moral paradox. So the legislative provisions of the Act stipulate that only permitted embryos may be used in reproduction, only permitted embryos may be transferred to a woman. And embryos become classed as permitted or non-permitted. I'll just draw your mind back to this complicated diagram of the origins, or the many possible origins of embryos, their many possible destinations, and what can happen to them in between. Embryos become classed as either permitted or not permitted, depending on their origin, their destination, and the process of what happens to them along the way. So the Act distinguishes between permitted and non-permitted embryos on the basis of those embryos that are produced from natural gametes, natural sperm and eggs, versus either synthetic gametes, modified gametes, or perhaps non-human gametes would also fall into the non-permitted category. It distinguishes between permitted and non-permitted embryos based on process. So embryos that have had cells added to them or have been genetically modified are excluded, they are non-permitted. And we know the exception that was created for treatment of mitochondrial disease that enabled the mitochondrial donation regulations. And of course, the distinction between permitted and non-permitted also applies with respect to the destination of those embryos, because the permitted designation applies primarily to transfer for reproductive purposes. So the putative paradox, if you like, the question is, what lies beyond the threshold of permissibility? If we think about embryos as liminal entities in the process of becoming, what becomings is the status of non-permitted embryo cutting off? What becomings are not permitted? I've often asked myself, what would actually happen after 14 days of development? What would happen if we allowed a non-permitted embryo to be transferred? Would we be creating something with moral status that we would be obliged to respect? Would we be creating something so monstrous that its becoming must be prevented? Or is what we are worried about is that we would be creating something monstrous precisely in its having of moral status, but in its being something that we are not prepared for, something that we don't expect? And so here are the hopeful monsters of my title: the embryonic becomings that are currently not permitted and that wait to become. And the question that I think, and I'm going to end with more questions than answers, is what possibilities for ethical evolution do these hopeful, not permitted monsters open up? What would happen if we crossed those bright lines that I talked about at the start, the 14-day rule, the heritable genetic modification? What would happen next? Well, in fact, we have already crossed that line when it comes to genome editing, and it was not the end of the world as we know it. There were, of course, serious consequences for science in terms of trust, in terms of self-regulation and scientific responsibility, and there were health and well-being concerns for the children involved. But these children are not monsters. The world didn't end. The monsters have not destroyed the world. What other possibilities might be out there when it comes to human and non-human chimeras? And I bring these up because I think they illustrate that our concern is about the moral troublings that such beings would create, and not the beings themselves. We might have experimental and welfare concerns both for the gestator, for women who might carry such embryos, and for the gestatee, for the chimeras themselves. Very tellingly, one of the primary responses to the idea of human-animal chimeras has been this idea of what Robert and Bayliss have called inexorable moral. Naturally, we would expect public reactions and consequences for science. And if we were to cross the bright line when it comes to synthetic gametes, synthetic embryo-like entities, or even embryos past 14 days, I think we would run into yet another paradox, in that we can't determine whether such entities, synthetic embryos, embryos from synthetic gametes, would in fact be equivalent to natural embryos without performing the very experiments that would be prohibited. So where does that leave us? This is my last slide. I can see I'm out of time. What are the implications of not only crossing the line, but in fact even thinking about crossing the line, raising the question of whether we should revise the 14-day rule, whether we should revisit the regulation of heritable genome editing, a question we're very likely to ask in the near future? I think concerns may attach to the idea of the slippery slope, and that we had one rule in place, and as soon as science pushed us to the line, we were willing to cross it. And I think we shouldn't look at it in that way. The regulatory line that exists is a fence. It may stop us from traveling in one direction. It gives us time to stop, to look over the fence and think whether we want to create a gate and open that gate. I think, in fact, a willingness to revisit rules, to look down what may not be a slippery slope, but decide whether we want to go that way, is, as I've argued, a necessary part of ethical evidence-based policies. I think that we need to, if you like, face our monsters. We need to confront the moral concerns over embryos rather than the morally concerning properties of embryos. So it's not so much about what such creatures might become as the moral concerns, the troubles that they would provoke in us. And finally, we need to revisit the idea of moral expertise for the 21st century. The Warnock Committee set a model for ethical policy making and the involvement of experts, stakeholders, and publics in a form of ethical policy making discourse. In the 21st century, I think it's become clear that our expectations for public involvement have changed, that there is a much greater expectation of involving publics in this discourse. If we look at genome editing, every report that's been published in the last 5 or so years has said we must consult with publics. But what hasn't been answered is, who are these publics? How should we consult them? How should that feed into ethical policy making? When should we do this? And so I think these are the crucial questions that remain to us to be answered. Thank you very much.